they did that. So again, essentially what we're doing is going in the opposite direction from the way it was really done, okay? So if you are interested in looking at patterns of gene regulation, you want to find mutants. So in this case, if we think about a completely normal cell that has a normal repressor gene, a normal operator, and a normal copy of LAC-Z, if we want to look at expression of LAC-Z, what would we expect to see if there isn't any lactose present inside the cell? Should LAC-Z be turned on or turned off?
Turned on.
Yeah, it should be off. And then when there is lactose inside the cell, what should happen to LAC-Z expression? On, exactly right. So if you are interested in trying to figure out how this happens, how LAC-Z is off when it's supposed to be off, and it's on when it's supposed to be on, you need to find mutants that don't do that. One or the other thing can happen that goes wrong. You can either have the gene being turned off when it should be on, or you could have the genes being turned on when they should be off. So some mutation causes expression to differ from what it should be, from what's going on in the wild type case. Okay. So actually at this point, let's talk about what's going to happen when we mutate these different components of the system. So let's imagine we make a mutation in the gene that codes for the repressor protein. So we essentially break this gene so that we don't produce the protein. If we were to do that, we'll just call that an I minus, meaning that we don't produce functioning repressor protein. How is that going to affect our pattern of gene expression? What's going to happen to LAC-Z expression if we do not make functional repressor protein? Yeah, it's going to be on all the time, exactly right. Okay. What about let's say our repressor protein is fine, but we make a mutation in the sequence that the repressor protein uses to bind to. So if we change this sequence so that the protein doesn't know how to bind there anymore, how is that going to affect our patterns of gene expression? Yeah, they're actually going to be the same thing. It's going to be on all the time. If you don't have the repressor protein, or if the repressor protein doesn't know where to bind because we've changed the sequence that it recognizes, we're going to get the same result. Now in this case, scientists refer to this mutation in the operator, a lot of times it's annotated with a C, which means constitutive mutation. What constitutive means is basically on all the time. So this mutation causes expression of LAC-Z all the time. Okay. So if you think about it, now we have a little bit of a problem now because we have two different mutations that give us the exact same phenotype. So how can we tell which one of these two genes is mutated? Okay, they give the exact same result in either case. So let's think about the strategy, or let's look at the strategy that the scientists used to try to investigate this, okay? What they actually did was create bacteria that had two copies of these genes inside of them, using those F' plasmids that we talked about last week. So let's go back to this case. Let's say our operator is okay. We have that mutant allele in LAC-I, so we don't produce our repressor protein. But now we bring in another piece of DNA that is okay. It has an okay version of LAC-I that can make repressor protein. has an okay version of LAC-O. And we can even simplify this a little bit further. Let's forget about LAC-Y for the time being. Let's just focus on LAC-Z. And this version is okay. So if you think about it, we have one copy of LAC-I that's not making functional repressor, but we have this copy that is making functional repressor. Let's imagine what we should see then in the presence and in the absence and presence of lactose. So the way I'm going to symbolize that in this chart over here is this slash is separating the two pieces of DNA. This is referring to this piece of DNA. This is referring to this piece of DNA. Yeah, question?
Is that the amerozygote?
Yeah, so the precise term for this is called a amerozygote, exactly right. So what do you guys think? What's going to happen now that we've brought in this extra piece of DNA that has a functioning copy of the repressor protein? How is that going to affect our pattern of gene expression here? So let's say our options, we have two options, either normal expression or constitutive expression. I didn't think to type this up on polls ahead of time, so we can just do it with a show of hands. How many vote for normal expression in this case? How many vote for constitutive expression in this case? Okay. Most of you voted for normal expression, and that is correct. If you think about it, as long as we have some functioning repressor protein here, it can bind to the operator of either piece of DNA, really, and regulate its expression the way we would expect. Okay, so what we see in this case is that it's actually the wild type allele of the repressor that's dominant, is another way to think of it. If I have a mutant allele and a wild type allele, the wild type allele wins. Now let's take this, let's do one more, let's make another mutation. So let's say we break this copy of LAC-Z, and so that the only copy of LAC-Z that can be expressed is the one that's over on the piece of DNA with the broken repressor. Do you think this repressor protein really cares which DNA molecule it's attaching to? It's floating around inside the cell. Yeah, it doesn't care at all, right? It's a molecule that can diffuse around, it can bind here, it can bind there. It doesn't care. So the fact that this dominant allele can come over here and regulate expression of this piece of DNA, the fancy term we have for that is transacting. So our repressor, our gene I, is transacting, what we would say, because it's coding for something that can bind to either piece of DNA. Why do we care? Okay, let's go back to what happens when we mutate the operator, and let's look at how it behaves differently from what happens when we mutate the repressor. So remember, when it's just the mutated repressor or the mutated operator by itself, we get the same phenotypes. Okay, so now what we're going to do, let me try to combine some of this. Let's say we go back to that original piece of DNA that has the mutated operator. So its genotype is going to be I plus, O, C, C plus. And then just like before, we're going to bring in another copy of everything that's completely normal at first. So maybe before we vote as a class, talk about it with with the people around you. What kind of expression pattern are we going to get? Are we going to get a normal expression pattern in this case? Or are we going to get constitutive expression when we change this sequence so that the repressor protein can't bind there? Even though there's another copy over here.
Express black seed all the time, right? What's transacting? What is science?
Show of hands, so how many of you vote for a wild type expression pattern in this case, where it's off in the absence of lactose, on in the presence of lactose? Okay, one. Couple. How many vote for constitutive expression in this case? Okay, the majority of the class. That is correct, okay? Because what happens in this case is that this piece of DNA here, if the protein can't bind to it, the protein can't shut down gene expression. And so this copy is going to be on all the time. Now again, what I was saying is that in this experimental system, this copy would be regulated okay, but we can't tell the difference. All we know is whether or not LAC-Z is turned on or not. So what we get in this case, this constitutive mutation, it's the mutant allele that's dominant to the wild type allele, which is the opposite of what we saw up here, right? Up here it was the wild type allele that was dominant to the mutant. So we have two different types of constitutive mutations. One that is recessive, the other one that is dominant. Now let's make another mutation now to look at what happens. Let's say we do this. We break this copy of LAC-Z that's on the same piece of DNA as that mutant operator. And then the other piece is completely fine. So again, talk with the people around you, predict what should happen in this case when we look at LAC-Z expression. So this copy is completely destroyed, so you're not going to get any expression from that copy. There's only one copy available to be expressed now. Okay, how many vote for constitutive expression? Okay, the majority votes for wild type, and wild type is exactly correct.
What did you do?
So would that, and why is that? Why would we see wild type expression in this case? We have a mutant operator over here, and we have, why is that? Why are we getting wild type expression even with a copy of a constitutive operator inside the cell? We have working repressor protein, but so did we up here, right? We had working repressor and a constitutive mutation. So what's the difference between, well, okay, so what's the difference between this and this?
The constitutive gene can't produce the LAC-Z.
So what you guys are getting at is the fact that this operator here is only going to regulate the copy of LAC-Z that's on the same piece of DNA that it is on, right? It's not going to be able to, if this protein binds or fails to bind to that operator, that's not going to have any impact on this copy that's on a different piece of DNA, right? It's only going to affect the copy that's on the same piece of DNA. The fancy term we have for that, the idea that you all look for, is that that operator is cis-acting. Okay. So transacting is a fancy way of saying that the gene can affect either piece of DNA. It does not have to be on the same piece of DNA as it is on. Cis-acting is the opposite. That mutation can only affect genes that are on the same piece of DNA. And so when researchers were investigating this system, this is what they found, is that they were able to isolate constitutive mutants. Some of them, though, that constitutive mutation was recessive, and the dominant allele could actually work on either piece of DNA, it was transacting. Other constitutive alleles were dominant, but they could only regulate the DNA that they were on. They were cis-acting. And so by trying to figure out what would cause those sorts of patterns, they came up with this model, that one gene coded for a protein that binds to a DNA sequence and turns on and turns off genes. Which to me is pretty remarkable that from patterns like this, they were able to make predictions like this that actually were borne out by once we did have the technology to clone genes and look at protein expression and things like that. Okay, let's do another mutation now. So we're going to mutate LAC-I again, but in a different way. Okay, what we're going to do is we are going to mutate LAC-I so that essentially lactose can no longer interact with the protein. The special term we have for this, we'll call it an IS mutation. I'm not going to tell you what the S stands for yet. And let's just ignore this other, I guess we should probably fix all this other stuff first. Let's get rid of this piece of DNA. So again, talk with people around you, make some predictions. If I make a change so that this protein can no longer bind to lactose, what sort of expression pattern are we going to see? Are we going to get wild type, or are we going to get constitutive, or are we going to get something completely different from what we have so far? Pattern we would see due to this mutation.
All of these things break down.
So if this protein can't bind lactose anymore, but it can bind DNA, how's that going to impact our pattern of gene expression?
We decided it would be minus because it's going to stay bound to the DNA regardless of the environment.
Or blocks the
Not
process.
allowed to.
Bingo. What we have is now a protein that is essentially insensitive to the environment. It's called a super repressor, which I think is probably one of the coolest names of mutation out there. So the S stands for super. Essentially what we have is a protein that remains permanently stuck to DNA. Okay, let's create our heterozygote again. Let's bring in our wild type piece of DNA and look at what happens now.
But as soon as he starts writing letters, everybody's like,
So who's going to win, in other words? If we have a wild type copy of the repressor and our super repressor, the name kind of hints a
Right now,
little bit.
Adam's
What do you guys, so talk about it with your partner. Now we have another cell, it's this heterozygote. We have regular repressor and we have super repressor. Who wins? Which expression pattern would we see in this case?
So tomorrow I need you to do something. And then Saturday, I'm gonna go for supposed to go to some blues thing.
My friends want to go to Fear Factory. If you want to go.
Okay.
Cool.
I mean, I hate serious stuff, but it'll be great.
I've never been to Thanksgiving, so
It's not like commercial for it's my
I'm trying to. I'll look at the text and see which one it was.
We really have three possibilities, right? We have normal, which is minus plus. We have constitutive, which is plus plus. And we have repressed, or we could just call it always off, I guess. So regular, always on, or always off. How many vote for regular in this case? How many vote for always on? How many vote for always off? All right, you're getting the hang of this, all
All
right.
I know
That's
is that
exactly
BYU
right.
kids
Now what we
are always
produce
on
is a trans
and always
active
repressed.
factor that can regulate either piece of, or combine to either piece of DNA, and it does so regardless of the environment that it's in.
What's that?
So again, you know, the neat thing to me is that we started with our model and predicted our genetic results. The way it was really done was the opposite. From genetic results, they built the model. But that's essentially how we worked out this system. So I'm actually going to skip through a bunch of slides because it's essentially
If your
working
penis
through the
goes
exact
in
same
the
thing we
vagina,
did on the board.
it's sex.
No, you don't.
Yeah.
Maybe my bishop doesn't understand, but I understand. Okay, perfect. Jesus, they should just do handies. Like, my God.
Well, yeah, so what you imagine, so let's imagine there is lactose in the cell. Okay, lactose would bind to this repressor and prevent it from binding to
You
DNA.
know, I've heard that, but I
But
think
this
it's called
protein that's floating
bagpiping.
around isn't able to be blocked, so it's still going to come in and bind to the DNA, even if this protein here can be prevented from binding. So one thing to bear in mind, you know, this is simple, we're
That's
just showing
amazing.
one molecule of each, but in the actual
This is going
cell,
on YouTube.
you're gonna have, you know, dozens of regular proteins as well as dozens of repressor proteins. And
Oh,
only
this is
two
what I
pieces
don't
of
learn in
DNA
genetics.
in the cell, so, yeah, it's not, yeah, it is a little perhaps deceiving in the way it's drawn, because it's not like this is only making one protein, and this one's only making. Okay, so that's the sort of the logic that we would work through in looking at gene regulation. There are other systems, so this was our negative inducible. There are other systems. We'll talk about a few examples, but we're not going to go as in detailed as we did with this guy. So the lac operon actually is under control of multiple environmental signals. Okay. What the operon wants to do is take lactose and break it down into simpler sugars like glucose. You only want to turn on those genes if you have lactose available. But another thing to bear in mind is that lactose isn't the only sugar that E. coli can use. There are other sugars out there that they can use. Some of them are actually easier to use than lactose. For example, glucose. If you are an E. coli cell and you have lots of glucose around, that's actually your preferred food source. So if you have both glucose and lactose, you don't really care about the lactose. You're just going to eat the glucose. And so glucose is actually another environmental signal that can be used to regulate the expression of these genes. But it does so in the opposite fashion. Remember, lactose was trying to turn the genes on. Glucose is going to help turn these genes off because we don't need them. So this is a case where glucose is acting as a co-repressor. This is our repressible part of the regulation. Now the way it ends up working out is it's kind of a several step system. There is another protein that interacts with the lac operon. It's called the catabolite activator protein, or the CAP protein for short. Unlike this protein that wants to shut off expression of the genes, this protein wants to turn them on. So this is where the positive comes in. So the CAP protein is a positive regulator. What ends up happening is that this protein needs a molecule to help it. It's called cyclic AMP. When you guys take cell biology, you'll probably learn a lot about this molecule. It's used in a lot of different processes. To make a long story short, what goes on inside the E. coli cell is that the glucose molecule prevents the buildup of this molecule that the CAP protein needs to turn the gene on. So, again, to make a long story short, if there's glucose, our activator protein can't bind and the gene is turned off. should have put another thing up there. If glucose is absent, then CAP can bind and our gene would help to be turned on. So this is kind of showing what's going on. If we don't have much glucose, we do have lots of cyclic AMP, and our activator protein can bind and turn on the genes. If we do have lots of glucose though, this protein can't bind, and so we don't turn on as much expression of these genes. So this is kind of neat because now we have, a system of genes that can input multiple environmental signals, both lactose and glucose. So if lactose and glucose are high, we don't get much expression. If they're both low, we don't get much expression. The only time we're going to get much expression of lac is if we have lots of lots of lactose and not much glucose, saying that we do have lots of this molecule available and we don't have anything better to eat, so we better turn on these genes so that we can eat our lactose.
I feel like that's like missionary.
One other example that we'll quickly walk through, again, not in nearly as much detail as we did with lac, is a trip operon. This gives us an example of another category. So we've done negative inducible. The CAP protein was our positive repressible. Here we have a negative repressible operon. What does the negative part refer to? We know just from the name what's going to go on here. We have our protein. What is it going to do to expression when it binds to the DNA? It's going to turn off the genes. And the repressible part, what does that tell us? The environmental signal is working in the same direction. It's also working to turn off the genes. Okay. So this is another system that the cell has to essentially prevent producing something that you don't really need. So these genes work together to help produce tryptophan. Okay, and if you think about it, there's only so much tryptophan you really need. Once you get to a certain point, you don't want to make more. And so this is the system that the bacteria have evolved to essentially decide when there's enough tryptophan. What is essentially going to go on here is that if we don't have a lot of tryptophan, we're going to make this repressor protein, but that repressor protein is not able to bind to the operator, and so we end up making these genes that can help us make tryptophan. As those levels of tryptophan get higher and higher and higher though, eventually they become high enough where they do start interacting with that repressor protein, and that repressor protein can bind to that operator and turn off those genes. It's essentially the cell's way of saying, hey, I've got enough of this tryptophan, I don't need to make more, and so I'm going to shut off expression of these genes now. Okay, so these cells have different ways of integrating these different environmental inputs to essentially decide whether to turn on or turn off gene expression. So what we've been looking at up to this point is essentially deciding whether or not to start the process of transcription, okay, the initiation of transcription. to protein. Bacteria also sometimes have another process known as attenuation, which is essentially deciding, okay, well, I've started transcription, but I really don't need these genes. I'm going to shut it down early so that I don't end up making these genes. So attenuation is a phenomenon that affects the continuation of transcription, not its initiation. This is another second level of regulation that actually works on our trip operon, the one we just discussed. And again, I think maybe rather than showing the slides, it may be a little bit easier to draw it out on a board. So one thing I will say is that attenuation is something that typically only occurs in prokaryotes, not in eukaryotes, and you'll see why in a moment. So let's say we have our DNA here, and we have these different trip genes. Forget which one was the first one. E. And then the others. Let's say that we've decided to start the process of transcription. So RNA polymerase has come in, it's bound to the DNA, it's racing along, and so let's say by now it is here. And so we have this messenger RNA that's coming out the back end of RNA polymerase. Okay, so that's our messenger RNA. One neat thing about bacteria is that because they don't keep their DNA inside the nucleus, they don't have to finish transcribing a gene before they can start translating, okay? So even while this molecule is running along and transcribing these genes, we can already have our ribosomes come in and start translating the proteins out of them, which is kind of crazy, right? So what's going to happen in this case is that part of this sequence has two very specific codons in it. It has two tryptophan codons. So remember, tryptophan is an amino acid. Now, let's imagine what's going to happen if there's not much tryptophan present inside the cell. Okay, our ribosome is going to run along. Now, what's going to happen when it gets here? It needs to plug in tryptophan to keep moving, but there isn't much tryptophan available. What's the ribosome going to have to do? It's going to have to wait. It's going to have to just kind of hang out for a while. Because that ribosome has to hang out there for a while, what ends up happening is that RNA polymerase is allowed to continue going on and finish transcription.
It's actually a sharper picture.
Okay. And so we end up making more tryptophan, which is a good thing in this case because we don't have a whole lot. Now let's imagine we have a situation where we have lots of tryptophan. Okay, so here is our TRPE gene. Here is our RNA polymerase. There's our messenger RNA. Oops. Here are those trip codons. Here's that ribosome. Here's our protein. If there is lots of tryptophan available, what's going to happen to our ribosome? Is it going to have to wait anymore? No, it's able to just kind of keep doing its thing. Now what happens is that because the ribosome can move so quickly, that causes a kink to occur in the RNA molecule up ahead of it, called a hairpin. And that hairpin is actually recognized by RNA polymerase, and it says, oh man, something's wrong. I better stop transcription. And so as a result, we don't end up making those genes that will make more tryptophan because we already have enough. So this is just a second level, a second system that the bacteria have to help regulate gene expression. It's sort of through this quirky way. It always kind of confused me, right? Because how can translation regulate transcription? Don't you have to do transcription first before you can translate? But the whole reason is because in bacteria, you don't have to separate them into different parts of the cell. You can start translating a molecule even before you finish transcribing that. And that gives you this opportunity for other levels of gene expression as well. Okay. So the slides do have some schematics of this. If this is not quite clear, I do admit it is kind of a confusing topic to think about. Most of the questions that are due on Sunday have to do more with the initiation of transcription. One thing we didn't get to a whole lot is how RNA molecules can affect gene expression as well. So I would just encourage you to look over the last few slides. It's pretty straightforward as far as what these different things do for the most part. So in some cases, you can actually have a different RNA that will interfere with translation of your messenger. That's called antisense RNA. Other cases, you can actually have a protein that will bind to the RNA and can affect how easily the ribosome can bind to it. Those are called riboswitches. And you can even have RNAs that will cut themselves into pieces under certain environmental conditions, which will, obviously, if you chop yourself up, you can't be translated then. And so that's another system that bacteria have to help regulate gene expression. Yeah. I can't remember what it's written on the syllabus. I don't actually remember whether this is the stopping point or whether we're going to do something else. I would look at this. Okay, guys, have a good fall break, and I'll see you back on Monday.
I'm studying. Well, a little bit.
I saw you
I
doing
was supposed to be
things.